Hello, my name is Nantia Leonidou and I'm a PhD student at the University of Tübingen in Germany. Today I will talk about tissue-specific reconstruction of constraint-based metabolic models based on ReconX. The human body comprises different cell and tissue types with different pathways. Thus, some pathways are essentially active in particular cells while inactive in others. For this reason, one needs to consider more context-specific reconstructions to study particular cell types. As you can see here, in tissue-specific models, reactions found to be inactive in the respective tissue, here highlighted with red, are removed from a generic model. The reconstruction of such models is based on either previously established biological literature-derived evidence or the integration of omics data and computational methods to constrain generic human reconstructions to the desired cell type. Those models are of great interest in cases of novel emerging viruses like the SARS-CoV-2, where they can be used to analyze metabolic changes in infected cells and detect targets which will inhibit the virus's growth. During the last decade, several algorithms have been published aiming the reconstruction of tissue-specific models from generic metabolic networks. Depending on whether flux distributions are predicted or not, these tools can be distinguished in two classes, as shown here. Our main goal was to reconstruct a tissue-related model and not only predict fluxes. Thus, we focused on the MBA-like approaches. These categorize the reactions beforehand in two sets, the core and the non-core, based on their level of expression confidence in the tissue of interest. Then they start pruning the generic human model by removing non-core reactions while trying to keep consistency in the core set, meaning that in the end no blocked reactions are included in the context-specific model. The algorithm mCadre uses a fully automated way to accomplish a tissue-specific reconstruction. However, so far it is implemented in MATLAB, and its functionality is based on the first version of the human metabolic network, namely ReconX, limiting its usability in the last few years since newer and more comprehensive model versions are available. Aiming to provide the community with an updated and more accessible version of mCadre, we re-implemented the algorithm in Python, leading to the new project called PyMCadre. Like mCadre, PyMCadre also reconstructs a single model in each run, using expression data and information of the network topology. The procedure still consists of three main parts: the ranking of reactions, the model pruning, and the consistency check. The ranking of reactions enables classifying reactions into core and non-core. To this end, it uses three scores: the expression-based evidence, which maps the ubiquity scores to the genes using gene-protein reaction rules, the connectivity of reactions within the model, and the confidence-based evidence, meaning the assigned confidence scores. The user has the opportunity to choose between FastFEA and FastCC to conduct internal optimizations and to check the model's consistency. The main goal of these functions is to detect blocked reactions and to deliver consistent networks by resolving a series of linear optimization problems. We also implemented scripts for data preprocessing along with the main code. Users can easily modify this depending on the utilized data to perform tasks such as obtaining confidence scores, binarizing raw transcriptomic data, and calculating ubiquity scores. To test the functionality of PyMCadre, we used ReconX as a generic human reconstruction combined with microarray data of human bronchial epithelial cells named BEC1. We also incorporated a biomass objective function to ReconX because it does not include one. The objective function originates from the human alveolar macrophage model published by Borpar et al. In 2010. Since we aim to use the model to detect possible antiviral targets against the novel coronavirus, we added a biomass objective function that maximizes the production of viral particles after infection. This pseudo-reaction considers the viral genome sequence, the subsequently encoded proteins, and their copy number, as well as the energy requirements for nucleotide and peptide bonds. The present table compares the results obtained from PyMCadre to the ground truth when FastCC was used for internal optimizations. As you can see, PyMCadre significantly reduces the time for creating tissue-specific models while maintaining the highest possible accuracy. Since we did not change the initial algorithm, this observation suggests variable performance of FastTC among MATLAB and CobraPy. However, with FastFEA, the resulted model was closer to the ground truth in terms of included reactions and genes in the pruned model, but at the cost of computational time. Also, the two pruned models shared over 1,100 reactions, meaning an overlap of around 82% of all reactions in each model. Thus, we have a substantial convergence between the approaches, establishing confidence for the quality of models generated by PyMCadre. Subsequent analysis of the MCadre integrated model allowed us to identify metabolic targets for antiviral therapies. As proposed by Aller et al., FBA and FEA can be used to predict key host reactions, especially in cases of novel emerging viruses. This can be computationally implemented in two different ways. Via single reaction deletions, where we investigate what will happen if a certain reaction is not allowed to have any flux at all, or using a less harmful for the cell approach called host-derived enforcement. Here the reaction fluxes are constrained to FEA-derived ranges, allowing the maintenance of the host optimal state while reducing the virus's growth. Thus, its reaction in the Recon1-BEC1 host virus model was analyzed using both approaches. Single reaction deletions in our integrated model revealed exactly one target enzyme that inhibits the viral growth, namely the nucleoside diphosphate kinase, or NTPK1. This enables the conversion of ATP and guanosine diphosphate to ATP and guanosine triphosphate. Its complete shutdown after setting both upper and lower flux to 0 reduces the flux of the virus biomass to 0 while maintaining the host biomass at 98%. And as you can see in this plot, no viral growth was observed after knocking out NTPK1. The enzyme NTPK1 was also observed as a potential target in the second experiment, the host-derived enforcement. This method, which Aller et al. Suggested, uses the results of FEA to recompute new flux ranges for all reactions and to systematically set individual lower and upper bounds. Targets were reported only when the viral growth rate with adapted fluxes was less than 50% of the initial growth rate. NTPK1's effect was found to be more assertive with a knockout experiment compared to the host-derived enforcement, because in this case there is no complete reaction knockout. Still, a small flux is allowed while inhibiting the reaction and keeping the host to maximum. Thus, further targets were reported. Moreover, among the reported targets, the guanylate kinase appeared, which proved to be a target in the human alveolar macrophage model, and it is also found in the purine metabolism like NTPK1. Its effect on the virus maintenance was observed to be weaker compared to nucleoside diphosphate kinase. All predicted reactions fall primarily onto the purine and pyrimidine metabolism, thus targeting pathways involving nucleotides to inhibit the virus could be of main interest. Please also note that inhibiting a reaction in a wet lab experiment within a specific range is much more challenging than a complete knockout. In the literature, potential inhibitors of the nucleoside diphosphate kinase have already been reported. For instance, a study from 2009 suggested angiostatin, polyphenols, and nucleoside analogues as potential inhibitors of NTPK, with polyphenols suppressing ATP production at higher potency than the nucleoside derivatives. Also, the Brenta database listed the first 3 compounds shown in the table on the left-hand side as potential inhibitors. Coronavirus belongs to the positive strand RNA viruses. Some of the already approved drugs targeting the action of NTPK found in the RegBank are already used for viruses of the same category. For instance, sofosbuvir is used against hepatitis C, or tenofovir and acyclovir against other RNA viruses like HIV and the herpes simplex virus. PyMCadre could be used to create models for diverse cell and tissue types. All these models could be then utilized to mirror interactions with bacterial pathogens or symbionts and the human as host, aiming the detection of effective therapies. Some validations could be done to improve the quality of PyM cadre. For instance, implementing automated test scripts will help to determine and ascertain the quality of the code. In addition, NDPK1 was reported as a very promising target to decrease the SARS-CoV-2 growth while maintaining the best optimal host state. So since compounds that may inhibit NDPK1 already exist, they need to be evaluated and verified in cell culture experiments in the lab. Also, as already mentioned, the host-derived enforcement outlined further candidate targets which inhibit the virus growth without harming the host by constraining their fluxes in a specific range. And these targets could be examined in more detail in terms of already existing drugs. Lastly, as a newer and more comprehensive version of the generic human metabolic model exists, namely Recon3D, PyM cadre needs to be computationally more feasible with extensive models containing a higher number of reactions. Also, tissue-specific models of other organisms except humans could be reconstructed and validated. The PyM cadre source code, as well as the test dataset and the resulted Recon1-Beck1 model, are stored in a repository found in our group's GitHub page. For more information, please visit our group's webpage or our YouTube channel. Thank you very much for your attention.